Okay, so let's begin talking about eukaryotic cells, chapter 5. Again, I've listed the objectives. A lot of this should be familiar to you because it was discussed. A lot of it was discussed in Biology 103 when we talked about eukaryotic cells and their structures. All right, so let's begin with the very first thing that we need to talk about was how did eukaryotic cells get here? We just spent a chapter talking about prokaryotic cells and bacteria, and now we want to talk about, well, how did our cells end up coming into existence? And one of the prevailing theories, and remember that a theory is not just an idea, it's got a lot of documentation, a lot of support that says this is how we think this occurred. Is called the endosymbiotic theory. And we take the word endosymbiotic, endo within, symbiotic, some kind of mutual relationship, and then theory, a lot of documentation that suggests that this is how it happened. So we know that prokaryotic cells were here first. Okay, duh, Earth was covered in water, we had prokaryotic cells here. But we believe at some point during that, 2 million years ago, that these prokaryotic cells took in smaller prokaryotic cells, and for some reason they began to act as a unit. In other words, they began to cluster together, and and instead of one cell having to do all functions, remember, metabolism, excretion of waste, stimuli, acquiring nutrients, reproduction, remember those characteristics of life, they began to parcel out responsibilities. So certain cells, certain smaller prokaryotic cells were kind of tasked with, okay, you do cellular respiration, you make ATP, or you have chloroplast in you, so you absorb the sun's energy and you make energy that way, or they began to divvy out responsibilities. And what we think is that over time these conglomerations or aggregation of prokaryotic cells began to work cohesively as one unit, and that's how our eukaryotic cells came into existence. Now again, that's called the endosymbiotic theory, and there's a tremendous amount of evidence that supports that, and I could give it to you. I could give you some of that evidence if you need me to, but basically that's just a brief history of how we think our cells came to be. In this unit, we're going to be talking about protozoans and algae. Those are going to be your protista. We're going to be talking about fungi. We're going to be talking about animalia, which is going to be like your helminths, your worms, and your arthropods. And then we don't really talk that much about plants, because typically plants don't give us problems, but the others do. So typically these are the eukaryotes that we're going to be discussing in this chapter. Here's an example of a eukaryotic cell. These are things that you should remember, and if you don't, you need to make sure you take the time to go back and study up on this, because again, this is why 103 was a prerequisite for 220, is because we expect you to know certain basic fundamental parts of a eukaryotic cell. So when we think about a eukaryotic cell, when we think about our cells, well, there's three major parts. You've got the outside, the external structures. You have the boundary, okay, and then you have the internal structures. So what we want to do is we want to actually start breaking that down, okay? So let's look at some external structures. Again, appendages. And just like we did with our prokaryotic cells, we want to talk about movement, okay, locomotion. And so flagella. And so the big thing is, well, what does the flagella do? Well, clearly the flagella allows for motility or allows for movement. So the question becomes, well, what's the difference in our flagellas, our cells that have flagellas, and bacteria's flagellas? And so the first thing you need to understand is it is a microtubule arrangement. It's a 9+2. I always laugh and say it kind of looks like a Twizzler shape, and it's thicker. It's like 10 times thicker than a bacteria's flagella, And another key feature that's different is instead of rotating or spinning like a propeller, it whips back and forth. So it tends to go left to right, thrashing more than in a circular rotation. So again, still does the same thing. It propels the cell from one place to another, but it just typically does it differently than your bacteria do. Okay, cilia. Cilia is one that we have on our cells, eukaryotic cells, that are not seen in bacteria. And so cilia is going to be like flagella, but they're going to be shorter. There's going to be way more. They're going to be very numerous. And they are going to do a more rhythmic stroke, for lack of a better way of saying it. The way I describe it is like the difference in rowing a boat versus having a propeller. Right, which is not a propeller, but rather than just having one oar, you've got a lot of oars in the water trying to move you. Again, they are found in protozoans and in certain animal cells. Typically, they're also used for movement, motility. They're used for feeding, and they're used for filtering. Okay, so the function of the cilia is for motility, for feeding, and for filtering. All right, from the appendages, let's move to another external structure called the glycocalyx. So the outermost boundary that comes in contact with the environment is called the glycocalyx. It is usually composed of polysaccharides, again, sugars. Polysaccharide means the sugar that's not typically easily broken down. And it is a network of fibers. It can be a slime layer or a capsule. And its purpose is not only protection and helping the cells stick, but it's also to help protect the cell. Okay? So if we think about the glycocalyx, it says fungi and most algae have a thick, rigid cell wall. Protozoans and animals lack a cell wall and only have a membrane. So again, not all eukaryotic cells have a glycocalyx. Okay? Cell wall. Again, provides shape. It's rigid. We're only going to see a cell wall in fungi and in algae. Okay, so we're only going to see a cell wall in fungi and algae. And fungi have a chitin. I've heard it called chitin before. I say chitin. A chitin or a cellulose composition, and that is going to be important because of chitin being an animal polysaccharide. So fungi are very similar to animals in the fact of that chitin, and I'll talk more about that in the next little video. Algae can be all kinds of things. Its chemical composition is varied. It can be pectin, it can be cellulose, it can be silicon dioxide, calcium carbonates. Their cell walls are all over the place. So again, I'm not going to spend a lot of time on that. Okay, membrane. Okay, typically it's going to be a phospholipid bilayer. Okay, duh, right? That bilayer, proteins, we expect that. The difference is that our cell walls are going to have, cell membrane is going to have sterols, right? And an example of a sterol that's in our cell membrane is cholesterol. Okay? Sorry, cholesterol. Remember that the membrane serves as a semi-permeable barrier. We know what that means. It allows some things in, keeps other things out. And we know that, we also know that eukaryotic cells are going to have a tremendous number of organelles in them, and each one of those is going to have a little membrane around it as well. All right, so here we go. Which part of the eukaryotic cell is responsible for contracting the outside environment, contacting the outside environment and signaling between cells? So which part is responsible for contacting the outside environment and signaling between cells? Okay, hopefully you said the glycocalyx, okay? The glycocalyx is the outermost portion, and that's the part that would actually signal chemically to other cells what's going on. All right, other internal features. Here's the internal features. Good heavens, lots of them, right? And I love this table because it's just a short, visual representation of the nucleus, the nucleolus, the endoplasmic reticulum, the Golgi, the lysosomes. Again, these are things that you're supposed to remember from 103. And if you're sitting there going, I don't remember this, then this is something you need to spend some time with. Okay, so let's talk with nucleus. All right, what's the big difference in our nucleus and the nucleolus region of a prokaryotic cells? Well, it's just that. We actually have a nucleus. Okay, and so the nucleus is going to contain chromosomes. So typically we have more than one, and inside of the nucleus there's going to be this dark area called the nucleolus, and this is where RNA synthesis occurs and ribosome assembly occurs. And we typically have our DNA in the form of chromatin when it's real thin and it hasn't divided yet, and then it turns into chromosomes when it begins to thicken up. And then remember, the chromatids is when it separates, and then when it comes back. So when the cell's just kind of hanging out and doing its thing, we call our DNA material chromatin. All right, then you have the ER, the endoplasmic reticulum. Think of this as highways. You have the rough and the smooth. The rough ER contains the ribosomes. The smooth ER doesn't. The rough ER is where the proteins are packaged and transported. Now, where are the proteins made? That's right, they're made in the ribosomes. But the ER is where they're fixed, they're packaged before they're shipped out. Okay? The smooth ER, smooth, no rough, so that means no ribosomes. This is going to be where lipids are made. And if you'll remember from 103, carbohydrates, lipids, proteins, and nucleic acids are the 4 macromolecules found in all living things. So the rough ER is identified because it's got the bumps on it, it's got the ribosomes, it's going to be really close to the nucleus. It is responsible for packaging the proteins. The smooth ER is also near the nucleus, but it doesn't have the ribosomes, and it's important for the production of lipids. Okay, Golgi apparatus. The Golgi apparatus is like the Walmart distribution center of the cell. What? What does that mean? Well, think about, what does the Walmart do? Or if you don't like Walmart, you could use the post office. It receives, it sorts, it packages, and it ships. So I think of like a distribution center because I picture the vesicles, the tractor trailer trucks pulling up to the warehouse. They drop off their supplies. Those supplies are then sorted. Okay, what do you mean sorted? Well, where is this going? Where's this going? Where's this going? It's sorted. It's then packaged. Imagine that saran wrap like material that goes around it, right? All of this is going here, so let's package it together. And then it's put back on another 18-wheeler, right, diesel truck, to be taken away. So it's like it's received, it's sorted, it's packaged, and it's shipped. So the Golgi is going to be kind of on the outskirts of the cell, and it's going to be responsible for collecting the lipids, right? Where does the lipids come from? That's right, the smooth ER. It's going to package the proteins. Yeah, remember who did the sorting of the proteins? That was the rough ER. It's going to take whatever is made, and it's then going to ship it either somewhere else in the cell, or it's going to ship it out of the cell to other cells. So that's what I think of when I think of the Golgi apparatus. Okay, and remember that the nucleus is going to give the code to make the proteins. The ribosome is going to make the proteins, but the rough ER is going to package it. It's going to send it to the Golgi, right? And then the Golgi is going to put it in little trucks, vesicles, and then those are going to take and secrete it to wherever it needs to go. Okay, so there's kind of a representation of that if you think about it. Nucleus gives the code to make the proteins. The ribosome parts are out here. The ribosomes are where the proteins are actually made. The rough ER packages it, gets it ready, ships it out. Look at the little transport vesicle. They pull up to the Golgi, and in the Golgi, They're sorted and packaged, and then they're shipped out of the cell. Okay, so that's a really great visual representation of that. Okay, lysosomes are also a vesicle, but instead of more of shipping, they actually have an enzyme inside of them that helps to digest food particles, and it can also, it contains acid usually, and it can actually help kill invading microbes. Vacuoles are like vesicles, but they're much bigger, and they're more for storage. Okay, they're more for storage. Don't really care about phagosomes. Okay. All right. And so mitochondria. So I'm going to stop right here, and I'm going to pick up with this in the next video because this one is talking about energy production.